Hello and good evening, buddies. How are you, everyone? I hope everyone is doing fine. And I am your biology mentor, Aafeeyat Ma'am. I'm here with one more episode of our series, which is Class 9, Term 1. Okay, just give me a thumbs up in the chat section that am I good to go or not. Let me also quickly have a look that am I clearly audible and visible so that we are good to go. Okay, we are good to go, I guess. Okay. So, moving forward with the classes, I just want to have a quick recap so that we can move forward with our classes. What have we studied in the last class? Okay, so let me give you a quick recap. In the last classes, what we have studied is cell is the basic, structural, functional and the fundamental unit of life. Yes, exactly, I'm talking about the name of our chapter. That is the fundamental unit of life. Yes. So there we have studied that how cell was discovered by Robert Hooke. It was dead cell. Then Leeuwenhoek. Then comes Robert Brown for the nucleus. Then came of course the cell theory, which was given by Skleden and Swan. Then comes Rudolf Virchow with the modification of that cell theory. Then comes multiple scientists with the invention of microscope. Yes, the moment microscope was evolving, that's how our everything, like everything regarding the discovery of cell and within the cell, because it's a microscopic structure. So with the discovery of microscope, everything was getting more discovered. So then we have studied about we have different types of shapes of cell. Then we have different types of cell shapes depending upon the function they perform. We have smallest cell, that is mycoplasma, largest cell, that is ostrich egg, longest cell, that is neuron. And we have studied about different shapes, that is irregular shape, which is not fixed boundaries. Then we have studied about the regular shapes, which has fixed boundaries. And then we had studied about the cell structure. Cell structure consisting of basically four things. Four things or four structures, that is cell wall, which is not in each and every cell, but then comes cell wall is present in plant cell, bacteria cell, that we have studied in the fungi cell. Then we had studied about the cell membrane, which is present in each and every single cell. It is not like that, it is not present in this, it is not present in that. Then we had studied about that how cell wall is formed. It's basically outer protective covering. Cell membrane is basically formed from lipid and protein. Then it's basically a protection or the barrier between the external environment of the cell and the internal environment of the cell. Okay, then we had taken forward the chapter with the nucleus, then cytoplasm. Nucleus basically brain of the cell, which consists of basically mostly important, like most important structure, that is the chromosome. Chromosomes are rod like structures which are formed by DNA and protein. Like the packaging of DNA and protein will form a rod like structure that is termed as chromosomes. Okay, then we had studied that these chromosomes are basically responsible for transferring the genes from one generation to the other generation. Okay, then we had moved forward we have one more viscous fluid which is present in the cell, which gives other organelles the space that they can move here and there without getting disrupted, without getting destroyed. That is cytoplasm, that is a cellular fluid. Okay, then we had studied about multiple cell organelles, which includes plasma membrane, responsible for the transportation. Transportation is basically the osmosis and diffusion. Osmosis, water movement; diffusion, the concentration movement. Other organelles, basically the gases I'm talking about. Then we had talked about we have different kinds of like different kinds of solution, different kinds of environment. Their cell act differently, plant cell act differently, animal cell act differently, because plant cell consists of cell wall. That's why the difference won't be visible through our naked eyes. But if I talk about animal cell, there you can see the difference. Yes. Then we had studied about we have cell organelles, which includes endoplasmic reticulum. Endoplasmic reticulum basically a membranous structure which consists of cisternae, vesicles and tubules. If ribosomes are attached to it, it is rough endoplasmic reticulum, which is granular, responsible for protein synthesis. If ribosomes are not attached, it is smooth endoplasmic reticulum. It is agranular, where lipid synthesis is going on. Okay, I have told you about the factory thing, that if anything, the factory needs the production. The production house is endoplasmic reticulum. After the production, there has to be key packaging and transportation of the products. So that is done by another cell organelle, that is Golgi apparatus. So Golgi apparatus is responsible for packaging and transportation of the goods or the material which has been already formed by endoplasmic reticulum. So endoplasmic reticulum is forming two things, that is protein and lipids. So the gift wrapper or the packaging will be in the form of glycogen, or the glycosylation process is going on. So that forms protein into glycoprotein and lipid into glycolipids. That is done all by Golgi apparatus, or we can also say at Golgi body. It was discovered by Camillo Golgi. Okay, then after this, Golgi body is also responsible in the formation of lysosomes and food vacuole. Now, I am talking about a factory, okay, I am talking about a proper industry. So, goods are formed, packaging, transportation, everything is done. Now, what will happen if I have more goods? What will happen if I have the distorted or the waste products? So, where will I dump those things? Where will I store the extra products? So, for the waste products, we have a specialized organelle, that is lysosomes. So, anything which is waste and which is causing harm to the factory and the goods, everything, that is distorted in the lysosome, because lysosomes are consisting of specialized enzymes, which is hydrolytic enzymes. So hydrolytic enzymes are basically which are where water act as a catalyzer. Water is pushing forward that enzyme that you have to act and you have to disrupt this structure. So hydrolytic enzymes are basically those enzymes which act in the presence of water. So what they do, they simply disrupt everything what is coming inside the lysosome, which is causing damage to the cell. Okay, now if I have extra goods, where will they go? They'll go to a storage house, and that storage house is known as vacuole. That we will study today. Okay, we had studied till here कि हाँ lysosomes are there, suicidal bags are there. So we had studied कि हाँ ठीक है, everything is there. Now we have to move forward with the energy. From where the energy is coming, because the factory is running on the power, that factory is running on the energy. So that energy is coming from a powerhouse of the cell, that is mitochondria. Yes, everyone who has ever studied science in their lifetime, they know about this word, which is mitochondria. So yes, I'm talking about mitochondria. Mitochondria is basically the powerhouse of the cell. So all the power, all the energy that is coming, or that helps in performing a cell its function, cellular activities, that is given by mitochondria in the form of ATP, that is adenosine triphosphate. Adenosine triphosphate is also known as the energy currency of the cell, because obviously you need money. So that is energy currency, ATP. So in the form of ATP, like what happens when the cellular activities or when the cellular respiration is going on, basically the glucose is broken down into carbon dioxide, water and energy. That energy in the form of ATP is used as the energy molecules or the energy currency. So that energy currency is utilized by the cell to perform its vital activities. Okay, then mitochondria is double membranous organelle that we had studied. It consists of its own ribosomes, it consists of its own cellular fluid, that is matrix, and it consists of its own DNA, which is responsible for protein formation also. So that's why it will also have a function apart from the energy generating, that will also be responsible for protein synthesis. Okay, now moving forward with our classes, we have multiple cell organelles coming, and the next cell organelle which we have to study, that is plastid. So plastids are not present in all the cell, in all types of cell. Plastids are basically present where any autotrophic movement or any autotrophic nutrition is going on. So plastids are mostly present in plant cell. We can see plastid in plant cell. So plastids are the main character, or the main role in the plant cell is performed by plastids. So plastids are basically responsible for multifunctions. Okay, multifunctions, like why am I saying multifunction? Because we have three types of plastids. Three types of plastids performing different functions. So three types of plastids are included as chromoplast, leucoplast and chloroplast. Okay, so chromoplast, basically the word chromo means the color. Basically the word chromo, this chromo means color. If I am talking about the chromo, I am talking about the color. Okay, so chromoplasts are responsible for providing color to the cell or any part. Okay, wherever this chromoplasts are involved, they provide color to that particular structure. If I talk about the chromoplasts are involved in the flower, they'll be providing or they'll be giving color to the flower. If chromoplasts are present in the fruit, they'll be providing color to the fruit. So wherever chromoplasts are involved, students, they are responsible for giving the color to that particular structure or the particular part of the plant. So chromoplasts are responsible for providing color. So these are colored plastids. These are colored plastids. If I talk about leucoplasts, so leucoplasts are colorless or they are white or transparent in color. So leucoplasts are colorless or transparent plastids. And if I talk about chloroplasts, so these chloroplasts are basically the green colored plastids. These chloroplasts are green colored plastids, as they are responsible for providing green color, and they're also responsible for performing a vital function in the plant cell, that is photosynthesis. That we'll study. Don't worry, we'll study chloroplasts separately so that we can have a good explanation about chloroplasts. So I was talking about chloroplast, leucoplast, and chromoplast, there I go. Okay, so I was talking about chromoplast, leucoplast, and chloroplast. So chromoplasts are basically responsible for providing colors. So what colors is it responsible for? If I talk about their functions, if I talk about the functions of chromoplasts, so chromoplasts are responsible for providing color. And what kind of color? It will be red, yellow, or orange color. So chromoplasts are responsible for providing red, yellow, or orange shade to that particular plant part. When I'm talking about leucoplasts, so leucoplasts are, the function of leucoplast is basic. These are responsible for the storage of fats, oils, proteins. Okay, these are responsible for fats, oils, and proteins. Okay, where to write? Okay, here only. So these are responsible for basically the storage. These are responsible for storage. Storage of what? Storage of fat, protein, and oil. Okay. So these are responsible for the storage of fat, protein, and oil. Okay, moving forward to the chloroplast. If I talk about the function of chloroplast, these chloroplasts are responsible for the very important function in the plants, that is, they perform photosynthesis. They perform photosynthesis, which is a food making process, and they are also responsible for providing the green color, because these chloroplasts are green colored plastids. So they are also responsible for providing green color. They are also responsible for providing green color to that particular plant. So if I'm saying that these chloroplasts are more in leaves, because they are responsible for food making process, which is photosynthesis. So that is the main reason why leaves and the stem is more green in color than compared to flower and fruits. Okay, as you have seen, the early fruit is also green in color, and later on stages it gets the color. So where chloroplasts are changing into chromoplast, that is the main reason for gaining the color. Okay, moving to the chloroplast, let's have a detailed discussion about the chloroplast. When I'm talking about chloroplast, okay, just give me a minute. So we were talking about chloroplast. Oh, we have lot of messages. Let me just have a quick look at the messages. Okay. Oh, we have answers also today. It's good to see that. Hello Aditya. Show notes on board, ma'am notes. Okay. Himanshi, notes, whatever I am writing, that is considered in the notes only. So you can have a look there. Okay. Sushant, it's not like she is not studying. She's studying. That's why she's asking for the notes. It's okay if she's asking. I don't have issues with that. Okay. Is my voice too slow? Am I not properly audible to you? Okay. Okay. Hello, Piyush. Okay. Hello, Sia. Once my voice is not clear, let me just have a look. Okay. I'll just have a look. Okay. What about now? Just check and let me know. Let me also check. Okay, I guess I am audible. See, don't waste the class time, okay, by doing such things. Okay, see, I am clearly audible. I can hear it. See, beta, just just increase the volume, turn on the volume, okay? Whatever device which you are using now, just turn on the volume. So you can hear me better. Okay, thank you Piyush for replying. Okay, let's move forward to the chloroplast. So I was talking about the chloroplast. This chloroplast are the structure. These are green colored structure which are responsible for food making process. So when I'm talking about chloroplast, the first thing, if I am talking about them, so the function is coming to my mind. So let's write about the function only, because we have already discussed this. Structure we will discuss in this slide only. Okay, I was talking about the function. Their function is basically to perform photosynthesis. Photosynthesis is a food making process in the presence of sunlight. Photosynthesis is basically a food making process in the presence of sunlight. Okay, so this is basically the photosynthesis, and these are also responsible for providing the green color to the plant cell. Okay, when I'm talking about the structure of chloroplast, I have already told you this later when we were studying about the mitochondria, that only two cell organelles are there, which are semi-autonomous cell organelles. Semi-autonomous cell organelles means they are performing with the cell, but they have their own functions also going in their cell body, in their cell organelle body. So this chloroplast also is a part of cell, but it also has its own functions. It's not like it does not have its own functions. So chloroplast is a cell organelle which is a part of cell, but it also has its own functions. That's why this chloroplast is also, let's change the color for this, is known as semi-autonomous cell organelles. Semi-autonomous. Okay, I am writing auto again. It's okay. We'll just erase it and then write again. Okay, semi-autonomous cell organelle. Okay. Moving forward with the structure, if I talk about the structure, I have already told you we have only two cell organelles, three basically. Three cell organelles which has double membranous structure, which is mitochondria, chloroplast and nucleus. We have already learned about the other two. This is the last one. Okay, so when I'm talking about the structure, it is a double membrane. So if it is a double membrane. Okay, do someone have the problem if I write 2x, or do I need to write, I'll write 2 only. What if someone says, ma'am, I cannot understand this 2x. Okay. So talking about the structure, it is double membranous structure. It consists of double membrane, means two membrane. One is the inner membrane and the other is the outer membrane. As we can see in the structure also, this is the outer membrane and this is the inner membrane. This is basically the outer membrane and this one is the inner membrane. So two membranes, outer and inner. Okay. Then coming inside the structure, it also has its own fluid. This cell organelle has its own fluid and that fluid is known as stroma. That fluid is known as stroma. Okay. Now, what does this stroma consist of? This stroma consists of a coin-like structure which is stacked one upon the other. So, this stroma consists of coin-like structure which is stacked one upon the other. And if I talk about only one single coin, this one single coin is known as thylakoid. Thylakoid. Okay. And this thylakoid consists of its own inner fluid. This own inner fluid is known as lumen. Okay, basically the membrane part. This is lumen. When I talk about the stacking of the coin, like one thylakoid upon the other, when I'm talking about like one upon the other, one coin, then other coin, then other coin, then other coin, then other coin. So when I'm talking about the stacking of these coins, this is known as grana. So one is known as granum, and like when I'm talking about all the stacking, I'm talking about grana, which is a plural form. Okay, grana. So this one granum is connected to the other grana by a bridge-like structure, and this bridge-like structure is known as stroma lamellae. Why stroma lamellae? Because this bridge is present in the stroma. That's why this bridge is known as stroma lamellae. So one grana, like this is one grana and this is my other grana. This one is my other grana. Okay. This other grana is connected with the help of this bridge. Okay. This bridge is known as stroma lamella. This is basically the connecting bridge. Between two grana. So this is basically the connecting bridge between two granas. Okay, when I am talking about this, its structure also consists of its own ribosomes. Okay, apart from this double membrane, it also consists of its own ribosomes. It also consists of its own DNA. As I have already told you, these are semi-autonomous cell organelles. So they have their own fluid, they have their own ribosome, they have their own DNA. So basically, these are running their own and with the cell also. That is why these are known as semi-autonomous cell organelles. Okay, so this is the structure of chloroplast. Okay, one more extra information which is not in your NCERT, but one more information. So what happens in the photosynthesis is we have two types of reactions, which is one is light reaction and the other is dark reaction. It does not mean ki light reaction in the presence of light and dark reaction in the presence of dark. No, it's not like that. Light reaction is basically which is occurring in the presence of light. But the other name what we have given, which is a dark reaction, that is not because it is happening in the dark. That is because we didn't had the other name. So what we did was, this is light reaction. Now opposite to light reaction is the dark reaction. So that's why the name was given light reaction and the dark reaction. So we have two kinds of reaction in the photosynthesis. So this photosynthesis, okay, let me change the color for this. So photosynthesis involves two types of reaction. This photosynthesis involves two types of reaction. One is the light reaction. Other is the dark reaction. Okay. So we have two specialized places for performing these reactions. Light reactions occurs in the thylakoid or the grana part, and the dark reaction is occurring in the stroma part, which is the proper fluid part. Okay, so we have two places. With one is thylakoid. Here we have light reaction. And if I talk about this stroma part, okay, when I talk about this stroma part, it is here dark reaction is going on. Okay, makes sense. So do I need to write there also so that it makes sense to you when you study later on? Okay, so light reaction in the thylakoid and this dark reaction in the stroma part. Okay, this makes sense now. Okay, so this is extra information for you. It's not written in your books. You, even if you don't memorize it, it's okay. But you should be aware that photosynthesis is occurring in two parts, light and dark. Light basically in the presence of light, but dark, it's not like it will happen in the dark. It can happen anytime. But after light reaction, the other reaction which is occurring, that is dark reaction. That's why we have named it so. Okay. So this is the structure and the characteristic of chloroplast. Moving to the next cell organelle, which is vacuoles. Okay. Moving to the next cell organelle, which is vacuole. When I talk about vacuole, these are the storage houses. Vacuoles are basically the storage houses. The storage houses. When I'm talking about any storage house, I'm talking about vacuoles. So vacuoles are the storage houses. Well, okay, we have two types of cells. We have already studied on the basis of the nucleus, on the basis of cell wall. There is plant cell and then there is animal cell. So we have vacuoles divided into two only. So when I talk about animals, animals have very small sized vacuoles. Animals have very small sized vacuoles. But when I talk about plants, plants has a large vacuole. Plants has large vacuole which takes about 50 to 90% of the cell size. Okay, let me just. Okay, so this is our plant cell. And this is a vacuole. This is a vacuole. And it takes about 50 to 90% of the cell size. So in plant cell, vacuoles take up to 50 to 90% of the cell size. So they are large and it can be central and it can have multiple vacuoles at a time. Okay. Moving to the plant cell, this plant cell, the vacuole of the plant cell is basically the single membrane structure. So this vacuole is a single membrane structure and this membrane of this vacuole is known as tonoplast. This membrane of this vacuole is known as tonoplast and it has some liquid filled inside it. And that liquid is known as cell sap. Cell sap, which includes amino acids, proteins and multiple components. Okay, which includes, okay, let me write down that also. The cell sap includes amino acid, proteins, carbs, fats, okay, etc. So this cell sap includes all these things. Okay, moving forward with the structure of vacuoles. These vacuoles are the storage houses. It is responsible for storing all the food material which has been formed or the other things which has been formed inside the plant. Plant has more bigger cell vacuoles than animal cell. Animal cell has a small size cell vacuole because they are not producing. These producers are the plant cell. That's why they have large vacuoles because they'll be producing the food. Okay, so producers has the large capacity to store the food and we are the consumers. So we'll simply go there and eat it. Okay, that's what we do. Now, when I talk about vacuoles, there are two types of vacuoles. I can characterize the vacuoles in two types. If I want to talk about vacuoles, I can characterize this vacuole in two types. What are those two types? First is the food vacuole. Which I was talking about so far. I was talking about the food vacuole which is present in plant cell and the animal cell. But we have another type of vacuole also. That vacuole, this is basically for the food storage. We have another type of vacuole also which is a contractile vacuole. contractile vacuole, and this contractile vacuole is present in mostly the unicellular organisms. Mostly unicellular organisms, it is present in mostly unicellular organisms. It is present mostly in unicellular organisms. Like, for example, how can I forget this example, which is amoeba. Of course, just after drawing the diagram, every one of you must be knowing the diagram which is of amoeba. So yes, I'm talking about the single-celled organism like amoeba, paramecium, euglena, those unicellular organisms. I'm talking about them. So these unicellular organisms, what they have is, they don't have the normal vacuole. They don't have the normal food vacuole. They have specialized vacuole, which is known as the contractile vacuole. Okay, what is the speciality of this contractile vacuole? So this contractile vacuole is not only responsible for storing food. This is also responsible for other things, like if I talk about the functions, because obviously this is a specialized mechanism what unicellular organism is having. So if I talk about the function, this is also responsible for expelling the waste material, intake of food, and one more important, that is osmoregulation. So now what are these three? Okay, when this unicellular organism are there, they do not have proper organ system, they do not have proper organ mechanism. That's why they need a specialized structure, so that that particular specialized structure can perform multiple and the various functions. So that multiple and various functions regarding to the food and the water and the maintenance of the body is done by this contractile vacuole. This contractile vacuole, with the help of water, it helps in the water flow. Okay, so when the water is flowing through this contractile vacuole, it also intake the nutrition, and then it also expels the waste product out from its system. And with this, it also helps in maintaining the osmoregulation, the concentration of water and salt in the body. So this is a specialized mechanism by contractile vacuole, which is present only in the unicellular organisms like amoeba, euglena, paramecium, that we already know the names. Okay, that's why I'm saying the names. Okay, now moving to the next cell organelle, which is ribosome. Okay, how can I forget very important cell organelle? So ribosome is the protein factory. Wherever this ribosome is coming, I should be remembering that this ribosome is there, that means it will form or it will synthesize the protein. So that is why this is also known as the protein factory. It is also known as protein factory because it is responsible for the synthesis of protein. That's why it is also known as protein factory. Now, talking about the structure of ribosome. Okay, first let's look at the structure. So when I talk about the structure of ribosome consists of two units. The bigger part is known as the larger subunit. When I talk about the smaller part, this is known as smaller subunit. So this is the structure of ribosome, which consists of two units. One is the larger subunit and the other is the smaller subunit. Okay, moving to the ribosome, I have two types of cells on the basis of nucleus, that is eukaryotic and the prokaryote. That is the reason I have two different types of ribosomes, which is 70S ribosome and 80S ribosome. Okay, first let's classify this ribosome. When I talk about ribosome, I have basically divided the ribosome into two types of cells, which is prokaryotic cell and other we have is eukaryotic cell. So this is the reason I have two different type of ribosome, because prokaryotic cell consists of different types of ribosomes and eukaryotic consists of different types of ribosomes. So when I am talking about a prokaryotic cell, obviously it will be little less modified, little smaller than the eukaryotic cell, because prokaryotic cell are what? They are the primitive cell, and eukaryotic cell are what? They are the new type of cell or the true cell. That is the reason prokaryotic cell has a less version of the ribosome and eukaryotic cell has the higher version of ribosome. That's what you can say. Okay. When I talk about prokaryotic cell, this prokaryotic cell includes 70S type of ribosome. This prokaryotic cell consists of 70S type of ribosome. 70S type. And when I talk about eukaryotic cell, they consist of 80S type of ribosome. Okay. Now, we have two units of ribosome. Okay, we have two units of ribosome. One is the larger unit and the other is the smaller unit. So, when I talk about the larger unit, this will be the ratio. Okay, now add. Don't come in between when I say that 70 is equals to 50S plus we have 30S. Don't add, because when you add it will be sum up to 80S. Don't add them. Okay, they are not additional values, they are just the approximation value when you do the sedimentation coefficient, when you do basically the sedimentation. After that, whatever the value is coming, that is this 70S, not the addition part. Do not add them. I need to add a note in this. Do not add them. So when I talk about the 80S, we have again two units. We have the larger unit which is 60S and the smaller unit which is 40S. Again, it will be equal to 100. Don't add them. Okay, I am warning you, do not add them. I need to add this warning, I guess. Okay, I need to add this warning, do not add. Where do I need to add the warning? Okay, here we can write do not add. Of units. This is a clear note for you. Please do not do that, or else you will be confused throughout. How does it come from? Okay, so when I talk about prokaryotic cell, it consists of two units, that is larger subunit and the smaller subunit, where the larger subunit is the 50S and the smaller subunit is the 30S. And when I talk about the eukaryotic cell, where the larger subunit is 60S and the smaller one is 40S. So that S, whenever I'm talking about this S stands for sedimentation coefficient. This S stands for sedimentation coefficient. Like whenever I am doing the sedimentation, as you all know, the sedimentation is basically the properly, whenever I'm shaking something and then I'll have a proper line or the covering, or I'll have that process. So settling down process is known as sedimentation. So if I jiggle both the subunits like 50 and 30 all together, then I'll have a proper sedimentation coefficient, a proper value of the sedimentation, the settling down value, that will be 70S. That won't be 80. That is the reason the 70S is written there. Okay, when I settle down this 60 and this 40, I will have a value that will be 80, not 100. This is the reason this 80S is the whole ribosomal unit of eukaryotic cell. Now, when I told you about mitochondria and the chloroplast, they have their own ribosomes. So, this mitochondria and the chloroplast, as they are the part of cell, they are included in the cell as their cell organelles. This is the reason this 70S ribosome, this 70S type of ribosome is not only present in the prokaryotic cell, it is also present in mitochondria and chloroplast. So, okay. So, when I told you about this, okay, we'll go back. Okay, so when I was talking to you about this part, they consist of their own ribosome. So here their own ribosome means 70S type of ribosome. But when I say this whole cell, whole eukaryotic cell is including 80S type of ribosome. Okay, let's learn it this way. Suppose this is my eukaryotic cell. This is my eukaryotic cell. Okay, this is a eukaryotic cell. Let's write it down. Eukaryotic cell. This is just for your information so that you don't get confused in between that. Okay, so this is your eukaryotic cell. If I talk about the ribosome inside this cell, these ribosomes are ATS ribosomes. Okay, now when I say that let's have a mitochondria, okay, this is a mitochondria and this is the chloroplast. This is the chloroplast. This is basically the chloroplast. This is the chloroplast. So when I talk about their ribosome, which color to take? Let's take the purple one. When I talk about the ribosome in them, in their structure, here you have 70S type of ribosome. Here also you have 70S type of ribosome. So understood, even in the eukaryotic cell, we have two types of ribosome, ATS in the cell, okay, 70S, which is inside the cell organelle, that means mitochondria and chloroplast. Okay, then this ATS will be associated with the endoplasmic reticulum and inside the cytoplasma, because endoplasmic reticulum is also the part of cytoplasma. That's why the upper part of the, like what RER is now, that RER, their ribosome is also present. The ribosome present there on the RER is also ATS type of ribosome. Okay, so here we had studied about all the types of cell organelle. So here the topic of cell ends, but from where do these cell comes from? So they comes from the pre-existing cell. This was the theory given by Rudolf Virchow. Rudolf Virchow said that all the new cells, okay, all the new cells arise from the pre-existing cell. All the new cell arise from pre-existing cell. Like if I say this is my pre-existing cell, this pre-existing cell will give rise to the new cells. This is the theory given by Rudolf Virchow, which says omni cellula e cellula. All new cells arise from pre-existing cells. Now, when I say this will be my mother cell or this will be my parent cell and this will be this both. This both will be my daughter cell. Okay, this both will be the daughter cells. Okay, so these both new daughter cells are arising from parent cell. Okay, now when I say about cell division is basically a process where the formation of new cell is going on. Cell division is a process. Cell division is a process of formation of new cells. Now, what is the need of this cell division? So, when I say the need of cell division will be growth and repair of the damaged, injured and dead cells. This is one need, and there is one more need for the formation of gametes. Okay, understood? So, this cell division is a process of the formation of new cells. And why do we need this cell division? We need this cell division for the formation of gametes and for the growth and repair of the damaged, injured or the dead cells. For all these purposes, we need the process of cell division. So, if I talk about cell division, is it that easy that one cell will be dividing into two? Is it that easy? No, that won't be that easy. Now, what is the process exactly? First, when I talk about the cell division, first there will be a normal cell. So, let's call it a mother cell. This will be our mother cell. This is our mother cell. Now, there will be growth happening inside the mother cell because I want to make one cell into two. So, everything will be multiplied, everything will be going bigger. So, first there will be growth. First will be growth. So, this will grow. This is growth. So, there will be growth at first. Now, what will happen at first? The first process, the first step of cell division starts with the karyokinesis. So, when I talk about the parts or the process of cell division, it starts with the division of nucleus, that is known as karyokinesis. So, there will be first step of the cell division, that will be karyokinesis. That means one cell will be having two nucleus. This is the process of division of nucleus. So here what happened is karyokinesis. Karyokinesis. This is the first step. This is our first step, karyokinesis. And after karyokinesis, this one cell including two nucleus will divide into two separate cells, that will be cytokinesis. Now the division of cytoplasm will be there and it will form two different cells, and that process will be known as cytokinesis. This is cytokinesis. This is our second step. This is cytokinesis. Now what is this karyokinesis? Karyo means nucleus and kinesis means division. This karyo means nucleus and this kinesis means division. And this cyto means cell and this kinesis means division. So with the help of this, you can easily understand that what does this karyokinesis means and what does this cytokinesis means. So when I want this cell division to happen, this one single cell won't simply come to this phase. It has to go through multiple phases in between. The first phase will be the growth, because I want to multiply the cell into two things, into two cells, into two daughter cells. Okay, let me write down here also. These two which is formed, these are the, okay, let's not write it down here. Let's write down here. These two are newly formed daughter cells. These are newly formed daughter cells. Okay, so first step will be the growth of the cell. Then there will be karyokinesis, division of nucleus. So one cell will be having two nucleus. This is my nucleus. Okay, this is my nucleus. This is also my nucleus. So one cell will be having two nucleus, and then this nucleus will divide, and that even the cytoplasmic content will divide, and that process will be known as cytokinesis. Then we will have two daughter cells. Okay. Now, when I'm talking about both these processes, so cell division is basically two-step process. One is karyokinesis, the other is cytokinesis. So this karyokinesis is also of two types. One is mitotic division and the other is meiotic division. One is mitotic division, other is meiotic division. What is the difference between these two divisions? I am saying this karyokinesis, this whole karyokinesis, it is divided into two parts. First is the mitotic division or the mitosis type of cell division, and the other is meiotic cell division or the meiosis. Okay, these are basically the two types of karyokinesis, how the division of nucleus is happening in a cell. So those are of two types. One is mitosis and the other is meiosis. Let's learn deeply about these two types. Okay. Okay. First is our mitosis division or the mitotic division. This division is also known as the multiplicational division. This division is also known as multiplicational division. Okay. This division is also known as multiplicational division. When I'm talking about multiplication, every time that reminds me ki I need to multiply the copy. I need to increase the copy. That is the term what multiplication means. Increasing the number, that means multiplication. So multiplicational division or the mitosis is the increasing number of the cell. So when I'm talking about, like when I have one cell, that will be forming exact replica. Exact means exact replica, but two replicas. Like when I want to have multiplicational division, I'll be standing like one Aafeeyat standing, and then I'll have two Aafeeyat, like one Aafeeyat standing here and one Aafeeyat standing here. So that is multiplicational division. Exact replica, it will be having same specs, same hairs, same facial structures, same height, same health, everything will be same. So one Aafeeyat and then there will be other Aafeeyat. So that is multiplicational division, forming the exact two replicas. I'm talking about two replicas. Mitosis division or the mitosis is the multiplicational division, which involves that one mother cell, will be forming similar or the exact two daughter cells, and these will be identical daughter cell. So what is mitosis? Mitosis is basically the process of formation of two identical, okay, spelling mistake, two identical daughter cells from single mother cell. This is mitotic division. कभी कभी ना स्पेलिंग में थोड़ी सी मिस्टेक्स हो जाती हैं, बट कोई नहीं, चलता रहता है. चलो आगे. Okay, I was talking about one mother cell dividing into two exact replica or two exact identical daughter cell. That is mitosis division. When I talk about the content of the nucleus consists of chromosome. What does a nucleus consist of? Nucleus consists of chromosomes. So when I talk about the nuclear content, the content of the nucleus, that will be chromosome, that will also gonna be same, like the same identical, same number of chromosomes is in mother cell, same number of chromosome in one daughter cell. I'm not talking about both the daughter cell. Like if I have, okay, let's take an example ki I have here 4. For example, okay, this is not for real, I'm talking about example. For example, I have 4 chromosome in the mother cell. Count. This is 1, this is 2, this is 3, and this is 4. I have 4 chromosome in the mother cell. Now count in the one daughter cell, like this is my first daughter cell and this is my second daughter cell. So this will be my first, second, third, and fourth. And now count in the second daughter cell also. This is first, second, third, and fourth. And you can see the color also. This is blue, red, one blue. Okay, now let's mark this also. 2 red, 2 blue. Same exact identical daughter cells. As I've already told you that exact identical daughter cells will be there. When I'm talking about the chromosome, so when I say 4 chromosome in the mother cell, then I will say, this also includes 4 chromosome. Okay, this also includes 4 chromosome. Talking about one daughter cell. Okay, so here I can write, okay, same number of chromosomes are transferred from mother cell to single daughter cell. I am not concluding both. If I include both, then it will be 4 and 8. Okay, understand this. I am not including both. I am just including one daughter cell. Equal number of chromosome in the parent cell and equal number of chromosome in one daughter cell. That is mitosis. Or multiplicational division. That is known as multiplicational division. Like here 4 and here 4. Multiples of 4 are, we got the multiples of 4, like 4 chromosomes in the one daughter cell and 4 chromosome in the other daughter cell. We got the multiples. Okay, now learning about the other type of karyokinesis or the other type of cell division, that is meiosis. Meiosis is also known as reductional division. Meiosis is also known as reductional division. Meiosis is also known as reductional division. Now with the word reductional, what do you understand? When the decreasing number of the chromosome is there, like I had the 4, now I have 2 chromosomes. That means reductional. Okay, now the chromosomes number are getting reduced. So when I say, okay, I forgot to mention the function, I guess, in this. Okay, we'll take it both at the same time. Okay, When I'm talking about meiosis, I will say ki, this is reductional division. The number of chromosomes are getting half. So when I say that this is a process, and this process, this meiotic division is occurring in two phases, meiotic first and meiotic second. So meiosis is basically the process of forming non-identical daughter cells, 4 daughter cells, okay. The meiosis is the process, this meiosis is the process of formation of 4 daughter cells, okay, 4 non-identical daughter cells. Let's write it this way. 4 non-identical daughter cells from single mother cell. From single mother cell. Okay. Now let's see where is the mother and where is the daughter. Okay. So this is the mother cell. This is our mother cell. Okay, this is a mother cell. So mother cell is consisting of some cell, okay, some amount of chromosome, okay, as we can see. This is our first meiotic division. This is our first meiotic division. That's why we also known as meiosis first. We also known this as meiosis 1 or meiosis first. So first meiosis, like after the first division of meiosis, we get 2 daughters. We get 2 cells. Now, after the second division of meiosis, we get second division of meiosis, we get 4 daughter cells which are non-identical. As you can see, here we have 1, 2, 3 and 4 chromosome. Okay, here we have 1 and 2 chromosome. Here also we have 1 and 2. Here we have 1, 2, 1, 2, 1, 2, 1, 2. So here when I say that I have 4 chromosome, when I am saying I have 4 chromosome in my mother cell, I have only 2 chromosome in the daughter cell. So that means it has been reduced like 4 to 2. So it has been reduced to 2. That is the reason this meiosis is also known as reductional division, from 4 chromosome to 2 chromosome. And now let's check, do we have identical copies or not? Okay. Here we have red and blue, blue and red, blue and red. Can you see red and blue too? Okay. Now when I check this first daughter cell, we have 1 blue, we have 1 red and blue, but we do not have blue and red. So this is not identical to the parent. Now let's check the second one. This is our first daughter cell. Okay, this is our second daughter cell. This is our third daughter cell, and this is our fourth daughter cell. These are all daughter cells. These are all daughter cells. Okay. So let's check the second one now. Okay. 1 red, 1 blue and red, but we don't have red and blue. Okay. We have here, we have 1 blue, we have 1 red and blue, but we don't have blue and red. Do we have that? No, we don't have that. Now let's check third one. We have blue and red, we have red. We don't have red and blue. We don't have red and blue. Okay, so these are non-identical daughter cell. How the mother cell was, these daughter cells are non-identical and chromosomes has been reduced to half. Talking about only 1 chromosome, okay? Talking about only 1 daughter cell, okay, not chromosome, talking about only 1 daughter cell. That the number of chromosome is reduced to half in daughter cells, DAU. Okay, again made the spelling mistake. Reduced to half. Okay, in daughter cell. For example, we have mother cell as 4 and now we have 4 mothers. Now we have 2 in daughter cell. Can you understand half? If I divide 4 by 2, it will be 2. That is why I am writing it is reduced to half. Now, what is the use of this cell division? As we have studied two use, growth and repair and formation of gametes. So these two types of karyokinesis, these two types of cell division, which is mitosis and meiosis, are working at different places. They have different area to work upon. Now, the area to work upon this mitosis is this, where? Okay. Where is mitosis happening? So when I say that mitosis is happening where we need growth, repair, where we need growth and repair, where we need growth and repair. Okay, where we need growth and repair and formation. What will happen there? There will be mitosis. There will be mitotic division or mitosis process. But when I talk about this meiosis, this will be, now let's talk about where in this also. This will be at the place for the formation of gametes, for the formation of gametes. Okay. Now we have two questions in our NCERT. Why this meiosis is happening in the formation of gametes? Because to maintain the variation in the life cycle, or to maintain the variation between the organism. If I say that I am the product of my mother and father, obviously they will be having different characters. So their different characters has been intermingled and been into me as a different character. So I do not have all the similar characters as my parents, but I also have the similar characters as my parents. So this is a conflicted statement. See, I have the characters which my parents have, both my parents, father and my mother. So I have their characters as well as I have the mix of their characters. That is the reason this meiosis is happening, for the intermingling, for the variation in between the generation to generation, and for the formation of gametes, because we need the variation, and that variation brings the continuity of species. So what does this, why in the formation of gametes? Because it brings variation. It brings variation. And why this variation is important? Because for continuity of species, for maintaining the continuity of species. This is the reason we need meiosis or the meiotic division. Meiotic division occurring in two phases, meiosis 1 and meiosis 2. Mitotic division only in one phase. There is no mitosis 1, there is no mitosis 2. Okay, only in meiosis division there is meiosis 1 and meiosis 2. Mitosis, multiplicational division, meiosis, reductional division. For cell division, first a cell needs to grow big, that is growth. Then there needs to be karyokinesis, which is division of nucleus, and division of nucleus can be in two types. So one is mitosis and the other is meiosis. Mitosis, then we have, okay, let's have a quick revision for this and then I can end up the class. Okay. So when I am talking about cell division, first there will be cell. Okay, this cell will be growing to big cell, and then they will have karyokinesis, like one cell will have two nucleus. Okay, when I am talking about one cell will have two nucleus. So we will have two processes. One will be mitosis and other will be meiosis. So when I am talking about mitosis, this will have one mother cell including two nucleus. How this two nucleus will be there? This one mother cell, okay. Here, this one mother cell will be forming two dot. Okay, let me say this has 4 chromosomes. This have 4 chromosomes. So this one daughter cell and this second daughter cell, this will also have 4 chromosomes. Okay, this will also have 4 chromosomes and similar chromosome. But when I talk about meiosis, how it will be there? There is one daughter cell. Just a quick recap. One mother cell is dividing into two cells first. This cell, this is having 4 chromosomes. This will have 2 chromosomes. This will also have 2 chromosomes. This will have more 2 chromosomes. One more daughter cell. Here, 2 daughter cell, 1 cell, and another daughter cell. This will also have 2 chromosomes. This will also have 2 chromosomes. Okay. understood. So ultimately the number of chromosome is similar, but what's happening, meiosis is happening in two different parts, and different parts is this one is meiosis first and this one is meiosis second. So at the end of meiosis second we have 4 daughter cells. So meiosis is like one cell giving rise to 4 daughter cells. 4 daughter cells. And when I talk about mitosis, here one parent cell giving rise to 2 daughter cells. 2 daughter cells. So this is mitosis and this is meiosis. And after karyokinesis, we will have the cytokinesis, where we will get 2 cells or we will get 4 cells. If we get 4 cells, that means the process is meiosis, and if we get 2 cells, that means the process is mitosis. This is there for today. So here we end with our chapter and the topics what the fundamental unit of life teaches us. So in this chapter we had studied about the cell, the organelles of the cell, cell theory and the cell division, because the cell division is an important part, and this cell division part will also be a big chapter in your 11th standard if you take biology. So you can study there with me only, of course, because I am your biology mentor. Okay, so you will be studying the same topics in your 11th standard also, but in a broad way. But I have given you the enough introduction about the cell division. So this is for today. What you have to do next is, we are done with the chapter. You have to do is go read your NCERT books. We had taken some little instances out from the NCERT also, but not more than that. Okay, just for the information and to make you understand in a better way. This is the reason I have taken some instances out from your NCERT. Rest everything is in your NCERT. Okay, so I would suggest after this lecture you should go and read the whole chapter, get well versed with the chapter. Don't skip even the summary and even the do you know part. Do you know part, activities, summary, everything is important. Okay, don't skip anything, and I will suggest you to solve the questions. And if you face any query, any questions or any difficulty, any doubts, anything regarding the chapter, please ping me in the chat section. Okay, this is my humble request. Study, because this is the reason we are here. Okay, so please study and focus more on your studies. Stay hydrated. Never go out of the subject. Never go here and there. Wherever you feel that you are distracted, focus. Just tell yourself, focus here. This is where I belong. I have to clear this now. And then rest of the things I can think later also now. So stay hydrated, study on, focus on your studies, prepare your own notes, your personalized notes, so that whenever you get time or whenever you have less time to revise, even in the 5 minutes, you can revise by your own self. So I would suggest prepare your own notes. Okay, after learning the chapter from the lectures, we have completed the lecture and the chapter. So if you have not seen the previous parts, I would suggest you go and see the previous parts also and complete the chapter. Then you can revise the chapter on your own terms and then solve the questions. From this book, from any other book, you can solve. Let me have a quick look at the chat section. What's going on? Okay, thank you. Okay, okay, beta, vacuoles and lysosomes are different. Vacuoles are the storage bags, and lysosomes are the suicidal bags. But they are the formation of Golgi apparatus only. Golgi apparatus is the organelle which is forming both of these structures, lysosomes also and vacuoles also. But vacuoles are responsible for storing, and lysosomes are the suicidal bags. Okay. They are responsible for the waste dump. Anything which is causing harm to the cell now, they'll be disposing that thing. Okay. Okay, grana. Beta, grana is, okay, let me just have a quick recap for the grana. Okay, beta, this is grana part. Okay. So when I was talking about the grana, okay, one single. So this chloroplast consists of stacking of coin-like structure. This one coin is known as thylakoid. This one coin is known as what? Thylakoid. And if I stack this one coin to the other, like this whole stacking is known as granum. Okay, singular form is granum. When I talk about complete, like all the granums in the all the granums and the chloroplast, then I'm talking about grana. So granum is the singular form and grana is the plural form. Okay. Plants store food in their vacuoles, and they have, apart from vacuoles, they have personalized structures also. That those are known as leucoplasts. Leucoplasts are also the part of plastids which are also responsible for the storage. Okay. Okay. Any more questions? हाँ बेटा मैं सारी चैट पढ़ती हूँ ठीक है चलो if you have more doubts, if you have more question, ping me in the chat section. I'll surely get back to you as soon as possible. Or and if you have doubts regarding the questions also, I would suggest you to ping me in the chat section from any other books also. I am okay to solve your doubts. I am here for that only. So I would suggest ping me in the chat section. I'll answer as soon as possible. Till then, my chapter is over, my topic is over. I am taking your leave, guys. So I'll see you soon in the next chapter, in the next lecture. I am your biology mentor signing off to you. Bye bye. Have a nice day. Stay focused on the studies. Bye bye.